So we wanted to know whether the chickens have what's the lowest concentration of mycotoxins that will not affect the production performance and that will not cause any, bacterial infections or subclinical diseases or affect the disease production performance.
Good morning, everyone, and welcome to the Poultry Podcast Show. I'm Karen Grogan, your host today, and I have Dr. Revathi Shan with us from the USDA Agricultural Research Service. Welcome to the show today. Dr. Reva works across the street from me in Athens in the Russell Research Building, which is one of the major USDA research centers in the United States. So, Reva, could you tell us a little bit about your background and how you got to the position that you're at with USDA today?
Yeah, it's my pleasure, Karen. I'm happy to be here and not talk about my research. It's really a great opportunity, this podcast platform. Absolutely. And I was born and raised in India. I have just realized that there are many similarities between my hometown and Georgia. My hometown is a poultry hub, and is India's egg capital. Basically they call this as egg city or poultry town. My hometown is the second largest egg producer in India. Coming back to my educational background, I attended my vet school after graduating from high school, but not because of my interest, actually wanted to go to medical school. However, I was unable to enter to medical school. Obviously, our second choice was veterinary medicine. So I attended TANVAS. TANVAS is a Tamil Nadu Veterinary and Animal Sciences University, one of the renowned veterinary institutions in India, where I got my BVM and Masters in Veterinary Medicine. And I got my PhD at the Ohio State University, animal sciences, majoring in poultry immunology, where I studied about that, whether the embryonic incubation could be a potential strategy to, modify the post-hatching immune response, improve the disease resistance kind of things.
Excellent. And who did you do your PhD under at Ohio State?
Yeah, I did my PhD with Dr. Lilburn. He is my supervisor.
Excellent. So what are you currently researching for ARS?
Currently, I'm working with mycotoxins. I joined at the Toxicology and Mycotoxin Research Unit in 2020. So my current research program is focusing on the adverse impact of mycotoxins on poultry health.
Excellent. So a lot of people, have heard of mycotoxins and we talk about them frequently. So just give us a little brief explanation of what mycotoxins are and how they're formed in grain products.
Okay. Basically, these mycotoxins are secondary metabolites produced by the fungi when they're growing on the crops. The poultry feed commodities, they are not only get contaminated with these mycotoxins at the feed, but also during handling time, storage, whenever the temperature is favored for their growth. Humidity and temperature are the major player here. So because of that, it's extremely difficult for us to get a clean poultry commodities that are free of mycotoxins. So far, we have 400 mycotoxins have been identified, but the common poultry feed commodities are contaminated with these major mycotoxins like aflatoxin, acrotoxin, T2 toxin, fusarium toxins. So the poultry feed is the major ingredient. Because of that, these chickens are getting mycotoxins.
And so you said the numbers are large. We have over 400. So how do producers, how do they go about knowing, clearly probably most ingredients that come in will have some level of mycotoxins. What does your research look at in terms of mycotoxins? Since we know that they're a known entity, the levels may vary. You may have certain batches of corn or batches of other grains that have higher mycotoxin content. So, what does your research look at in terms of these mycotoxins?
Basically, when we think about this chickens, 65 to 70% of the poultry diet comes from corn. So we are using corn, soybean meal-based diet. So if you think about corn, soybean meal-based diet, we use 55% corn comes from, goes to the chicken feed. Some extreme cases, we use 72% corn. So all the major poultry feed commodities contaminated with either single or multiple mycotoxins. In addition, we do have the masked mycotoxins, so that's the different story. But my research is focusing on what is the lowest combination of mycotoxins that will not affect the poultry production performance, because the poultry feed ingredients not always comes with single mycotoxins. So co-occurrence of multiple mycotoxins give them a synergism or additive effect. So we do have the regulatory guidelines for these cytotoxins. Now, the FDA, they defined a certain level of mycotoxins in the feed. So for example, if we talk about that fusarium, fumonisins, that's the maximum permissible level is 50 parts per million or 50 milligram per kg diet. Okay. If we take that deoxynivalenol, it's a 5 milligram per kg diet. So that's the maximum permissible level. But if you take the chicken feed, it's not just containing one mycotoxins. So co-occurrence of multiple mycotoxins, they may have an additive or synergistic effect. So we wanted to know whether the chickens have what's the lowest concentration of mycotoxins that will not affect the production performance and that will not cause any, bacterial infections or subclinical diseases or affect the disease production performance. So in addition, I'm also interested in identifying the biomarker, real-time mycotoxin exposure and toxicity. Currently, we don't have any ideal biomarkers.
Right.
I'm also working on that area.
It's hard to test in the real bird. We can test in the feed and get quantities there, but we have no idea what's going on in the chicken. You had mentioned something called masked mycotoxins. What do you mean by masked mycotoxins?
Basically, masked mycotoxins, either amino group or glucose or sulfate group, that will attach to these major mycotoxins by biological or chemically and modify their toxicokinetic properties. Here, I just want to give an example. For example, fusarium fungus, fusarium verticilliaris or fusarium graminearium. These are toxins that commonly infect the plants when the plant grows in the field. These fusarium species produces toxins like deoxynivalenol. This is the normal process. What would happen if the plant has its own resistant defensive mechanism against these fungi? For example, the plant does this by attaching glucose molecule to DON. DON here is a deoxynivalenol. It converts this DON to DON3 glucoside. So now DON is in the form of DON3 glucoside, which is not toxic to the plant anymore. So the plant can survive without any problems. When we harvest this corn and analyze for this DON, we cannot find the DON anymore because DON is in the form of DON3 glucoside. But these masked mycotoxins, has an impact on the poultry. How does that happen? Because these intestinal digestive enzymes, particularly amylase, that cleaves the glucose molecule away from the DON within the GI tract if the birds ingested this masked mycotoxin contaminated diet. When we analyze the finished diet using the HPLC, we cannot detect this DON. because we don't have the standards for DON. We do have the standards for DON, but that will not detect the DON 3 glucoside.
Right.
So that is the problem with that with masked mycotoxins. So this is just one example. We have hundreds of masked mycotoxins for all major mycotoxins, like what I mentioned before. So the bottom line here is that we cannot detect this masked mycotoxins with conventional analytical.
Their current analytical technology.
Exactly.
Yeah. Maybe your research group can come up with a way to try to detect this.
Yeah, we are hoping so.
Okay. So just to kind of package that all together and tie it in to make it important to people joining in with us. So that is a protective mechanism by the plant, so it's not getting the negative effects. But then once it reaches the chicken GI tract, that extra glucose is digested off, and then it's back to regular DON. And DON is not a friendly player in the chicken intestinal tract and among other places. So what are some of the major health impacts that we see with these mycotoxins?
Yeah, most of the time, we know the negative impact of these toxins, because in the previous literature, if we look at the literatures, we know that the mycotoxins causes immunosuppression. We see the lesions on the liver, kidney, weak erosions, and in the GI tract we see that, especially in the gizzard, ulcers. Those if we look at the literature, some of them they use unrealistic dose. We don't see really in that common poultry ingredients. We don't see that level of mycotoxins. Sometimes the numbers will be really low, high. The one thing, biggest challenge we have that the testing and interpretation of that our lab results. That's the one thing. The second thing is that we have sometimes, in the producers, they use the strips, and strips, right? So they use that strip. That will work only with corn. If you are using the testing strip, using corn, only works with corn. That matrix will not detect that mycotoxin in the soybean. Yeah. So we have to use the different type of matrix. That's also gives some kind of error in our data. Next, that the clinical mycotoxicosis in chicken. Chickens, they have the broiler chicken, especially 42 days life cycle. When they feed the subclinical doses, they don't have enough time to show that real clinical symptoms like fatty liver or beak erosions, because they don't have enough time to show that, the symptoms. So poultry production standpoint, we really focus on that gut microbiome, immunity, and nutrition. These are in my perspective, they are like puzzle pieces. They have to fit together. They are connected to each other. At the same time, no one piece of puzzle is more important than other. All three of them are equally important. But in coming to this impact of mycotoxins, these toxins are targeting that gut epithelial cells or immune cells. These cells are highly proliferating cells with a high protein turnover. So basically, these toxins targeting those cells by inhibiting protein synthesis. So obviously, we see the symptoms related to gut epithelial cells. These are all the cells exposed to these toxins at the highest concentration. Mm-hmm. So as a result, that intestinal epithelial cells are kind of gatekeepers. They create a barrier between the lumen and the rest of the body tissue. So these toxins target those junctions and letting that pathogenic bacteria, toxins, everything enter into the blood circulation. As a result, we see that in worst cases, we see that leaky gut syndrome. Right. So we can see some kind of pathogenic bacteria enter into that blood circulation. Sometimes this may responsible for onset of certain infectious diseases like necrotic enteritis.
Right. It's definitely
necrotic enteritis.
One of those long lists of things that we know contributes to necrotic enteritis. I think we have multiple stresses on the GI tract, and mycotoxins are definitely one of those contributing factors. In terms of your research and looking at the various levels, at what level are the mycotoxins present to create that sort of intestinal tract damage? Are you talking about very high levels or very low levels, or like you were talking about earlier with sort of the synergies of multiple mycotoxins being present?
Yeah. Our goal now wanted to see that subclinical level of mycotoxins. We have that level by the FDA, but now in the poultry, ingredients have more than one mycotoxin. We wanted to go down a lot less than that recommended level or the tolerance level. To see the effect of these mycotoxins. So we evaluated the role of these toxins during the development of necrotic enteritis. So the question we had was that the damage caused by the fumonisins was big enough to cause necrotic enteritis, even without that coccidial pathogen.
Okay.
So we used 15 times less than the maximum tolerance level recommended by the FDA. So we used fumonisins is 3 ppm and deoxynivalenol we used 4 ppm. So it's a combined doses of subclinical level.
Those reports would come back that's low, low levels of mycotoxins.
Exactly.
So the two together, just so that I understand what you had the fumonisin and the DON at what we would all think is a fairly low level, single digit parts per million. That's not what you read in the books, and you could create clinical or subclinical necrotic enteritis without a cocci challenge, just with the mycotoxins.
Just with the mycotoxin, yes. So what we found that chicken diets with this subclinical level of these toxins decreased the body weight gain by 23 grams. We started noticing at day 14,
Day 14.
And this day 14. And this trend continued until day 35. These groups, the human dawn alone group, decreased the body weight gain by 84 grams. This is approximately 10.7% of the body weight loss, just because of this human dawn.
Just because of the no other differences, just the mycotoxin level.
Mycotoxin level. So,
and diets equal and everything else was the same. No other challenge between your groups.
Nothing. Just a
10% body weight difference.
10 points. Yes.
Yeah. And in terms of sort of evaluating and your research, is that from the intestinal tract damage?
Yes. We did that. We also, this is just body production performance wise, we saw that 84 gram differences, 10.7% difference body weight. And also we saw that 3 points in feed conversion ratio, In addition to that, we also looked into that intestinal histomorphology. So the mycotoxins damage the tip of the villi, obviously. So we also quantified that tight junction proteins. It's also decreased in response to these toxins. So in my opinion, there is no safe or unsafe level of mycotoxins. Low to medium level, it causes damage to the gut epithelium. So these are all the reasons. But we still these low level of mycotoxins, we are underestimated. It causes some effects on that bird.
Right. So in terms of, I think the industry accepts these low levels. So in a frame of your research, what are your recommendations based off of your research? What sort of measures could our companies start instituting to try to deal with these medium to low levels?
Yeah. So currently we ran a trial with a different combination of mycotoxins, starting from somewhere from 33 ppm of fumonisins with, 4 ppm of DON. And we gradually decreased that concentration up to 0.05. 0.5 of fumonisin to 0.6 of DON, something like that. But whenever the numbers were below 1 ppm of this fumonisin and DON, we didn't see that differences on that body weight.
Okay.
So, but no, I just presented this data in the IPPE, and so we need to go down a lot,
Right.
especially with the number of mycotoxins in the feed.
So in terms of the products that we have, there's mycotoxin binders and different clays and things like that that can be added to the feed. Is your research looking at that type of things? What measures we do have to try to decrease the mycotoxins?
Yeah, no, I haven't go into that level because first we wanted to know that whether this subclinical doses causes impact on that gut epithelium. But we had done previously a quite a few experiments with the Biomin. They have done a lot of research with these binders. But the problem with the binders are that we have the charcoal, for example, charcoal is that a century-old substances.
Yes.
These are really good with good strategy to get rid of the toxins. Mm-hmm. But the problem is with that, they are really working well with aflatoxin because aflatoxins, they have the planarity. So they can be able to go and sit with that clay, because the clay, they make different layers. They can be able to go and sit. You can go and park your car wherever you want in your assigned spot like that. They can go and sit. But when we think about this fumonisin and DON, they don't have that kind of planarity. So they cannot sit well with this binders. So our other option is to find kind of using some kind of enzymes. For example, microbes, so they can secrete some kind of enzymes. They can metabolize these kind of toxins into non-toxic metabolites. But they market currently they have some kind of options, but they are a little bit expensive. Right. So we had to come up with some broad spectrum, economically feasible kind of alternatives.
We're in the industry of making, pennies on things. So people need things to be economical additives. In terms of, you were just mentioning that the microbes, so how do these mycotoxins affect, what we understand in terms of like the GI microflora and the microbiota and all of that? What role are they playing there?
Yeah, when we think about these chickens, the microbial breakdown, it happens only at the ceca after passing through all the primary absorption site, jejunum, ileum, and then they reaches the ceca, and the ceca where you have the tons of microbes, they will break down that. But by the time these mycotoxins reaches the ceca, especially the DON and fume, they have damaged the small intestine. So in terms with chickens, the benefit of this microbial degradation is very, very minimal. So we wanted to know whether this chicken microbiota have the ability to detoxify the toxins. So we wanted to know what would happen, with these mycotoxins. But we have tons of questions in front of us.
So that's the fun of research. It generates questions, right?
Exactly. So we don't know the real impact of gut microbiota and mycotoxins, especially in chickens. And the effect of mycotoxins on gut microbiome, we don't know. And the third question was that we don't know the health impact on the host if there is a gut microbiome profile changes due to mycotoxins. So what we did, in our previous research, we found out that how this chronic exposure to subclinical fume and DON affect the composition and diversity of the fecal bacterial microbiota in chickens.
Okay.
So, what we had found out that these toxins, subclinical dose, again, this 3 ppm of human, 4 ppm DON, it changed the gut microbiome profile, starting from the phylum level to family level to genus level. For example, if we take the proteobacteria, at day 21, these mycotoxins significantly decreased that proteobacteria relative abundance by 0.8-fold. Not only on that proteobacteria, but also on that Lactospiraceae and also that Lactobacilliaceae. When you think about the Lactobacilliaceae, they are very good at detoxifying mycotoxins.
Mm-hmm.
But what happened in my research that the Lactobacilliaceae relative abundance was completely decreased, and it was replaced by the Clostridium. So
our friend Clostridia, it's always there. It just takes one thing to tip the scales.
Exactly. So we thought that, okay, there's no swapping between this proportion of this Lactobacilliaceae versus this Clostridium. Maybe involved with chickens are unable to detoxify these mycotoxins. Maybe this is the reason that create intestinal dysbiosis, reason for subclinical necrotic enteritis. But still, this is the very beginning. We still have long way to go to understand the gut microbiome, to get the clear picture of the impact of mycotoxins on gut microbiome aspect.
So your research is just still starting in terms of that aspect, but it's like it definitely does have some impacts along with what it's doing with direct damage to the intestines.
Yes.
So you had mentioned in your research you use some different. You were talking about like tight junction proteins. So these are pretty high tech types of tests to look at intestinal damage. What could you see as a test kind of in the future that you had talked about? We don't really have a test for in the chicken what the mycotoxins are doing. We can test the feed. What are some predictions that you might that you're working on, or you could have a current research plan in terms of this might be something worthy of looking at? So
currently, we don't have I'm really interested in looking for and identifying some biomarkers. Currently, we don't have any readily available biomarkers that would help to determine the real-time exposure of mycotoxins. So far, the markers we have for especially the human DON are after the fact, based on decrease in production performance. Right. And currently, what we are using that is the spingonin-sphingosine ratio, especially this fumonisins inhibit the particular ceramide synthase, which is part of that cell wall.
Okay.
So basically, this inhibit that particular cell wall mechanism. So we see that spingonin-sphingosine ratio. That is the one, but this one is also after the fact. But I wanted to see whether I can be able to find a real-time exposure of mycotoxins using certain biomarkers. So I am working towards on using some microRNA biomarkers.
Okay.
Something that. So I'm working on that. Maybe in next 6 months, I may have an answer for that.
That would be great.
Towards, we'll have
to check back in and see, or catch you at another meeting presenting the research for your lab. Good luck on trying to find that magic biomarker.
Yeah.
So in terms of chickens are presented with feed that contain a mycotoxin. It doesn't sound like they really have your work is showing that like they. The microbes are so far down the intestinal tract and the ceca that they don't really have any defense mechanisms.
Yeah.
So what, and I think your research has really shown that it does have, even at what we think are medium to low levels, really probably low levels, it's still having major impacts and disrupting the microbiome populations. And in terms of, where you see your research headed, you're going to search for this biomarker. What would be what next steps are you taking?
I'm going to know my next step. Now, we wanted to know whether this first step, we wanted to identify that low level of mycotoxins that will not affect production performance and also, no effect on the gut health. So whatever the research I have done with just to know one species, we used that Ross chicken. And so we found the data only on the Ross chicken. I wanted to see whether the same effect, whatever I seen with the Ross, to see it on another thing, whether cob, whether I can see the same data. And also I wanted to explore with the turkeys and ducks to see that impact. And the next thing is I wanted to identify the broad spectrum of mycotoxin detoxifier. Right now we have either enzymes, microbial enzymes, which is a little bit pricey, and then the other binders are adsorbent. It's not working well with this fumonisin and DON. Yeah. So I wanted to identify the broad spectrum anti-mycotoxin solutions economically feasible to the producers. So that's my next goal.
I'm sure everyone would appreciate that. No one wants to lose feed conversion, and we're constantly in our production systems. We're heading more and more towards no antibiotics ever, and limiting antibiotic use. So necrotic enteritis is always a constant challenge. So here you have given a much greater understanding of another important player in what is causing and contributing to necrotic enteritis. It's not just us removing antibiotics. It's these toxins that are present in the feed. And they're definitely playing a role. Your research is definitely needed. So the industry thanks you for doing this great work in mycotoxins. It's definitely a topic that people talk about, and I think that we just, like you said, there's 400 different metabolites out there. It's just a big scope of work in terms of trying to figure out which ones to research and what impact they're having. So you are starting a great career in a really broad, topic. And I think that, so that's great. Well, excellent. Well, thank you for sharing your breadth of work right now. And congratulations on presenting that at IPPE. If any of our listeners are interested, can they contact you at ARS? I'm sure you guys can find Dr. Revathi that way through ARS and through her abstract at IPSF. Thank you for your time today, and enjoy the rest of your day.
Thank you, Karen. Thank you so much for your time.
Thanks.
I appreciate that. Bye.
Bye-bye.